Good morning to all students. We are here with an interesting topic of industrial microbiology. Now, in the previous semester, we had microbial metabolism. Now, this time, I'll be teaching you this part of industrial microbiology. Now, basically, up till from first year to this year, we have seen that microorganisms plays a very vital or important role in generation of different kind of products, and these products are helpful or necessary for their own benefits and for the benefits of others. Means for what we can say, let us suppose if milk is there, then the milk by a process of fermentation gets converted into curd or any other useful products like paneer and so on and so forth by fermentation, and then it will be helpful for our consumptions. So if I am having, let us suppose, one organism and it is producing 10 milligrams of fermentative products, so that will be only a limitation of that organism to produce it for me or for a specific person or an individual only. But when it comes that we want to commercialize that product and we want that product to be given to an industry, and that industry would produce it by a larger scale in terms of fermentation, and then it will be provided to an n number of persons or to the entire society. So for that purpose, we need to understand and we need to study what is industrial microbiology, because we are, as on date or till date, when we are working in the laboratory, we are only working on a very small scale, and when it comes in terms of industry, we want that organism to produce that product in a very higher amount. So for that purpose, we need to understand what is industrial microbiology, and this is the paper in the last year 6th semester paper of industrial microbiology. So what is industrial microbiology? It is a branch of applied microbiology. Of course, applied microbiology in the sense that organisms produces some or somehow it produces a fermentative products, and that products will be applied product, and therefore it will be a branch of applied microbiology. It refers to the use of microorganisms in commercial enterprises, like what I told you just now, that these organisms are now being converted into commercial part, and these produces those commercial products in a very large amount, so that those can be sold and it can be reached to the society. The microorganisms are processed under controlled temperature, pH, to produce valuable products. Of course, now when we are dealing with industrial microorganism, industrial microbiology, it has to be under a particular protocol or a particular frame. Moving further. Now, the history, how this particular era started. As earlier 6000 BC, the Babylonians and Sumerians used yeast to make alcohol. During the First World War, British scientist Chain Weizmann developed a fermenter for productions of acetone. 1952, Hindustan Private Antibiotic Limited Pune made the first pilot fermenter in India, and Pasteur developed a new way of approach to industrial microbiology. So, industrial microbiology is not a very new branch, it has been practiced from a long period of time. Now, developmental phase in industrial microorganism, relative ancient, involves fermentation of alcoholic beverages and breads. Large-scale cultivation of yeast for brewing and baking industries, known as fermentation industries, where such mediums were introduced. Involves the batch culture of microorganisms. For example, penicillin production is one of the best example, where people used to produce the penicillin in a very large amount. Use of microorganisms as the bulk source of protein for animal and human for called as single cell protein, SCP. There is extensively nowadays also it has been extensively utilized. And biotechnology, recombinant DNA technology is applied in microorganisms for developing useful products. So why microbes? Now question arises, if there are industries which are producing a number of products, but why microbes are being used for production of such kind of things? So for that purpose, microbes are the mini chemical factories, possess a broad variety of enzymes, high metabolic activity, so it can grow and multiply rapidly. So microorganisms which are producing valuable products such as enzymes are being helpful for generation or for production of this particular kind of thing. Possesses a large surface area for quick absorption of nutrients. grow in inexpensive substrate. Byproducts of reactions will be substrate of another. Enabling the genetic manipulation should not be pathogenic or humans around. So these organisms are said to be, why they are said to be important in, or why microbes are to be utilized in fermentation industry. The reason behind the same is that these microorganisms will be helpful for generation of the product in a very simple amount. Very cheap amount of raw materials are being utilized for the production of say very higher or valuable products as well from microorganisms. So fermentations in a very basics, and what do you mean by fermentations? Chemical conversion of sugar to acid, ethanol and gases. Fermentations are done in a large vessel called as fermenter. This is most important, fermenter, because this part we have to deal a lot in the industrial microbiology part. So microbes are grown under controlled temperature and pH. Science of fermentation is called as zymology. Here this is most important. Zymology. Enzymology have a part, and zymology is a different part. Science of fermentation, when we are dealing with fermentation, and scientific achievements in terms of fermentations will come under the branch called as zymology. So fermentation steps. So what are the different fermentation steps? Any sort of fermentation it may be. It may be like production of beverages, or it may be like production of antibiotics, vitamins, so and so forth. So steps, somewhere somehow the steps are plus minus. In some reactions the steps may not be there. Some additional steps may be involved in this. But this is the general structure of a fermentation steps. Fermenter selection, microbial strain selection, fermenter media selection. So what sort of media has to be utilized for development of which sort of product that has to be utilized. Fermenter process, upstream processing, downstream processing means the extraction of the particular product. Quality control and assurance, of course, to maintain the quality the industry always looks in for. Product recovery and packaging. So these are all basic things that for an industry to run. These things are a very basic need. Unless and until you have these basic 9 points, what I told you just now, that some fermentations may have some different steps as well, which are in addition to these. So this is the very basic protocol of any sort of fermentation industry, wherein it has to undergo with all these steps, and then and only then the final product will be packed and necessarily be transported. A bioreactor differs from a fermenter in that the former is used for the mass culture of plant and animal cell instead of microorganisms. हा लक्षात घ्या, हा फरक बऱ्याच वेळा एक्झाम मध्ये पण विचारला जातो, डिफरन्स बिटवीन अ बायोरिएक्टर अँड ए मायक्रोऑर्गॅनिझम्स और सॉरी द फर्मेंटर. बायोरिएक्टर एक गोष्ट लक्षात घ्या, बायोरिएक्टर is generally utilized, I'm not telling you each and every time, but it is generally utilized for the mass production of mass culture of plants and animal cells. Means whenever there is a word called as fermentation industry is utilizing eukaryotic organisms or higher organisms, then the word will be bioreactor. But when it comes in terms of microorganisms, specifically bacterial or yeast or fungi, then it will always be a fermenter. The fermenter and the process, if you see the particular part of a fermenter, the fermenter have a different, various different gadgets and all sort of things. These are impellers. We are going to learn this in a very greater detail. Today only we are going to just brush up things of regarding industrial microbiology. Components of a fermenter: vessel to carry out the process, impellers, proper mixing, heat transfer prevents sedimentations, sparger, device that introduces air into the medium. See this now. See this, impellers, cooling jacket. If you exhaust is there, culture broth, the motor for movement, sparger you can see here, the sparger it is, the air bubbles you can easily see over here. Baffles to prevent the vortex and improve the aeration efficiency. If you see the baffles around the two ends of that fermenter. Temperature control, pH control and feed parts to add the nutrients. So fermentation media, what sort of fermentation media has to be utilized in this particular context? Carbon source, generally wheat or rice as a carbon source. Instead of direct sugars, we can utilize yeast of wheat or rice. Nitrogen source, yeast extract, one can easily utilize. So what are these upstream processes? Upstream process मध्ये काय काय घ्यायचं? Substrate, water, carbon, phosphate, nitrogen source, total media. It has to be sterilized and then it has to be poured inside the fermenter. Inoculum separates inlet air. Main fermenter मध्ये टाकलाय. Exhaust gases has to be removed out. And downstream process means harvest, precipitation, crystallization, centrifugation, drying, and the final product has been ready. So the process of downstream processing has to be very large as compared to upstream processing of the fermentation industry. See this production tanks, how big these particular fermenters are or the reactors are. In a process with large volumes of nutrient medium is inoculated initially. This is batch fermentation. About 2 or 4% microorganisms are added initially. After fermentation, microbes are removed and the material are isolated. Growth phase, lag phase, exponential phase, stationary phase, death phase. See this. In continuous, the medium is added continuously to the tank to replace that which has been fermented in the same. Now, this is most important. Remain in the logarithm. The organisms in continuous fermentations are remaining in logarithmic phase only. Chemostat is to prevent constant flow, keeping microorganisms in the logarithmic phase of growth. Turbidostat instrument measures the turbidity of the organism population to indicate the level of the growth. Fed batch fermentation, substrates are added in increments as fermentation progresses. Critical elements are added in small doses and it continues during the production phase. Production of antibiotics, study of antibiotics began. So we will stop here for today itself. So 3 fermentation processes are there: batch, continuous, and fed batch. In batch fermentation, the microorganisms and the final product has been removed. In continuous fermentations, the microorganisms are cultivated or the process of fermentation goes on in a continuous manner. In fed batch, the process, the substrate are added as the fermentation progresses very slow and slowly. Then and only then the reactions would take place. So that's all for today. In the next lecture, we'll try to continue with a specific product, for example, penicillin, in order to work. Thank you.